lecture, so I'm sure you understood many things, but if you have questions, just stop me anytime and ask. Right, so we'll just go over some interesting concepts that were brought up in homeostasis. If you can't hear me at some point or you can't see the screen or some glitch is happening, please let me know so then I can try to fix it, okay? So, right, what is homeostasis? So, as explained in coursework, it's essentially where it's a property of the system which allows it to be capable of maintaining internal conditions without too much variation despite external environmental changes. For instance, if we step into the cold or we step into a hot environment, we're able to regulate our internal body temperature such that it doesn't vary too much because we also function at certain optimal physiological conditions and set points, as was explained. So our body temperature is, roughly 37 degrees. So we want to keep it that way, and we have different mechanisms in place that allow us to maintain it at that. Obviously, if someone goes into an excessively cold environment for too long, that could result in a disbalance of this homeostasis. So you shift your equilibrium and your body starts getting colder than normal. So people can, in those cases, the body starts prioritizing what to keep warm and what to keep cold. So for instance, people who get frostbite, why does it happen, right? Why does frostbite start at fingers, toes, the tips of the nose? Because blood starts getting diverted away from those regions, from the extremities, because the body prioritizes keeping organs warm. So your internal organs warm in comparison to your extremities, which is why people who are in the cold for too long, maybe because of, some extreme sports or hiking or just an accident or something, they actually can develop frostbite, which can cause their fingers to lose circulation of blood, which can be highly dangerous and can lead to loss of those appendages. So that's an example of loss of homeostasis, or not loss, but just another way in the body is trying to maintain homeostasis under an extreme condition that can result in harm, even though it's keeping you alive, right? Anyway, that was just an extreme example. Most of the time, we're all pretty comfortable. Right. So, yeah, so as I mentioned, homeostasis is very important property for maintaining equilibrium in the body during your day-to-day activities or when you're adapting to some unusual environmental condition, right? And there are many different things the body regulates. So it maintains homeostasis with respect to temperature, as I explained, pressure, osmolarity, which I'll come to, and blood sugar and other things. And oh yeah, and one of the important rhythms the body maintains that helps in maintaining these homeostatic mechanisms is the circadian rhythm. So I'm sure all of you have heard of the circadian rhythm before. It's the natural cycle of physical, mental, and behavioral changes. And the body goes through this in a 24-hour cycle. So this is the human body. There are some animals that are not exactly 24 hours, and that's a very fascinating and not very well understood reason as to why. So circadian rhythms are most affected by light and darkness and are controlled by a small area in the middle of the brain, right? So they can affect sleep, body temperature, hormones, appetite, and many other functions. And if you have abnormal circadian rhythms or some work or some habit that causes the circadian rhythm to become different than normal, then that could lead to problems like obesity, diabetes, so on and so forth. So the circadian rhythm is often called the body's clock. And if you think about the molecular reasons as to why, actually there are, like, how is the circadian rhythm maintained? How does the body know when it's going through a 24-hour cycle, right? Because you can't tell your cells to regulate things in a certain way. So they have to be actual chemical detectable signals. So the body uses positive and negative molecular feedback loops as a mechanism to regulate the expression of genes that are called clock genes. So these are some examples mentioned here, and these can actually be regulated, and they can control the transcription translation of different genes and then downstream mechanisms, which which can then activate different kinds of cellular pathways and signaling pathways at different times of the day so that the circadian rhythm is maintained. And part of the circadian rhythm, right, a very important part of it is diurnal rhythm in the case of human beings. So humans are diurnal, we are diurnal, and in that means that essentially during the daytime our activity is heightened, and during nighttime there's relative inactivity, and so we are called diurnal. And the timing of an activity by an animal depends on the variety of environmental factors, of course, the temperature, ability to gather food, maybe, with respect to animals, the risk of predation, the time of the year, so the season, and so on and so forth, that can affect diurnal rhythm. And so a little bit of trivia here. We are diurnal, humans, monkeys, so on, we're all diurnal. But animals that are active during twilight, so early dawn, they are called crepuscular. This is something I also learned. I did not know this, or I didn't remember this. And those active during the night, as you well know, are nocturnal. So bats, owls, even mice are nocturnal. And animals active at sporadic times, so during day, night, and all, are cathemeral. So an example of cathemeral is a very well-known domestic cats. They are active both in the night and day, and they sleep sporadically and are active sporadically. So these are some examples, which I thought would be interesting for you to think about, because within the circadian rhythm, that is constant, but the diurnal rhythm is just a subset of that that tells us when we're sleeping, when we're active. And then that talks about the sleep wake cycle and the cycling of different types of molecules in the body, like hormones in the body. So the sleep wake cycle, you have in diurnal rhythm, you have alertness peaking during the day, and you feel sleepy, and the um, you have melatonin. Sorry, yeah, and you have melatonin peaking at night, right? That corresponds to feeling sleepy. You also have, so as I mentioned, hormones like melatonin high at night, cortisol, which helps with wakefulness, so that peaks early morning. And interestingly, growth hormone is secreted mainly during night sleep. So getting good night sleep when you're young can help with growing better. Um, Disruption of this sleep wake cycle can occur with people who work in shifts, nurses, people who work in different plants and stuff. Jet lag, when you travel, irregular sleep schedules, or if you look at screens a lot, especially at night, that can disrupt the sleep wake cycle, leading to problems in your circadian patterns and health issues as well. So, these are some tips to maintaining a healthy sleep wake cycle, which I hope that all of us can follow, right? And alongside the sleep wake cycle, part of maintaining circadian rhythm and part of what the diurnal rhythm looks like for humans is body temperature. So, body temperature, as was mentioned in your coursework, it fluctuates, right? So, it's the lowest in the early morning, it peaks in the late afternoon to early evening. So, this is all within a range around the set point temperature, right? And metabolism, of course, you have better glucose tolerance and insulin sensitivity during the day. And that's why they say don't eat late at night, because your digestion and energy expenditure are lower in night hours in comparison to daylight hours. So maybe there's a higher chance of deposition instead of metabolism if you eat like a late night snack. So just something to keep in mind, though we don't always follow it. Right. So another concept that was taught to you, I'll just go over it quickly here, which was osmolarity or osmotic concentration as it's called. So it can also be osmolality, which is Your milliosmoles per kg and milliosmoles per liter is osmolarity. Right, and it's a measure of the total concentration of osmotically active particles, essentially solutes, in a solution, which is expressed in these different units. And osmolality can actually be measured in the serum using osmometers. Um, and yeah, so even a slight alteration in water homeostasis in the body can have a profound effect on the cell size and function. So, maintaining water in the body, there's a major hormonal component to it, right? The thyroid is very important, as well as obviously the kidneys, and there's a detection of osmolarity that occurs with respect to ionic concentrations and pressure, and these things can regulate osmolarity in the serum. Right. So, so different cells, depending on their role and space restriction, have varying capacity to accommodate such osmolar stress. Right. So the central receptors, which are osmoreceptors, so you have different kinds of receptors. So this is the one that senses osmotic concentration. These are primarily present in the anterior hypothalamus in the brain. And as was mentioned in your coursework, there is a homeostasis in osmolarity. So the intracellular and extracellular are the same, which is 281 milliosmoles per liter. And if this were to change, then you would have water moving from a lower osmolarity to a higher osmolarity. Or what I remember how they taught it to us in school was that water moves from its region of higher concentration to lower concentration. So water's concentration, meaning that the solute, if water is highly concentrated, would be less concentrated. Yeah, so I hope that I didn't confuse you. If anyone has any questions, please just let me know. Okay. Right, and quickly moving to glucose homeostasis. So as we know, I'm sure either you yourself or family members have done some blood sugar tests. So sometimes they say, okay, you have to fast for 8 hours. So you fast overnight and then you go and you give your blood and they check um, what the level is of glucose in your blood. And the typical normal range for healthy individuals is roughly 70 to 100 or 99 milligrams per deciliter. Deciliter is nothing but 100 ml. Um, and that's normal fasting. So you've fasted for at least 8 hours here. And then they sometimes ask you to then eat a meal and then come back after 2 hours after the meal and they take your blood and they check it. So that meal, that sometimes they write it as PP on the slip. So that PP stands for postprandial. It just means after eating. So the normal blood sugar level, obviously it's going to be higher than what it is after fasting because you've eaten something, food has gone in, it's been broken down, and then glucose has entered into your bloodstream. So normal levels, and obviously insulin has also been secreted to sort of handle this sudden rise in glucose. So 2 hours after eating, this postprandial blood sugar level should not exceed 140 mg per deciliter. And so how is this regulation taking place in the body? Oh, sorry. So just in brief, in the pancreas, which is an extremely important organ for glucose metabolism, we have two types of cells, beta cells, alpha cells. You also have delta cells, which secrete somatostatin, but let's not talk about them. Beta cells in the islets of Langerhans, they detect blood sugar level increasing. So glucose level is increasing in the blood, and accordingly they release insulin. So insulin exocytosis takes place in these cells. So glucose enters them via GLUT1, GLUT2, I think, receptors, and then there is some signaling which leads to insulin exocytosis. And so insulin enters into the bloodstream and it starts to break down and convert glucose, right? And so that's what regulates the level of glucose post eating. Now, if you go for a period of time where your glucose level actually decreases, like you don't eat for some X amount of time after your meal, and the level decreases such that your alpha cells, which are also in the islets of Langerhans, they can detect this decrease and release glucagon. And this is a mechanism. So alpha cells are not functioning on their own, so they're regulated not only by the level of glucose in the blood but also by beta cells. So insulin itself can inhibit alpha cells from releasing glucagon. So there's some internal signaling. So it's not just glucose detecting, there's also some paracrine signaling going on here as well. Right. So that's a very sort of short explanation into glucose homeostasis. Okay. So now I have some questions that I made. I hope you can answer them. So please just either you can switch on your mic and answer which of the options you think is the right answer. Or if you want, you can also, if you don't feel like doing that, you can send the message in the chat and I'll read it out and then we can discuss. So I hope that's fine with everyone. I hope you do that, even though there's not too many people. Uh, right. So the master circadian clock in the mammals is located in which brain region? So this was not covered in coursework, but I wonder if any of you know about this. Anyone? Okay. So the answer was given B. Does anyone else have any other answers? So actually, the master circadian clock region actually is in the suprachiasmatic nucleus. So the answer is C here, and it's interesting how this SCN region works. So it's really close to the eyes, right? It's suprachiasmatic nucleus, so it means it's just above the region where the optic nerve goes. So it actually gets an input from there. So um, when we sense light and dark, right, with the help of rods and similar cells in our retina. So there is an input to the suprachiasmatic nucleus from the eyes um, that allows it to essentially understand whether there's light coming in or is it dark. So that um, so that helps us discern with the help of inputs from the retina as well as from the suprachiasmatic nucleus, and it helps us regulate the circadian clock. Let me just check one thing. I think someone's trying to join. Just a second. Um, okay. Okay. All right. Sorry about that. All right. Let's start. Well, let's go to the next question. So, which part mentioned here of the brain secretes melatonin? So, do you have the answer to this? To those who have joined just now, you can either message the answer in the chat, which option you think it is, or you can unmute yourself and speak out the answer. Yeah. Anyone, any idea? No? So melatonin is the hormone that helps regulate sleep and wakefulness. So it makes you sleepy essentially. So it peaks at nighttime and then its secretion wanes during the day. So the answer here is actually option A, which is the pineal gland. Okay. So this was mentioned in the coursework as well, I think, by the professor. So it's good to know. All right. So next question. Okay. So melatonin secretion is highest at which time of the day in humans? So I think I've given away this answer already. Anyone? Yeah, right, exactly. Night. Great. So, yeah, that's one set of questions. Let's move to the next. So, which of the following best defines a diurnal organism? So, we just discussed what a diurnal means, also what nocturnal is, what the other uh, the other terms are. Yeah, exactly. Great. So, yeah, a diurnal organism is active mainly during the day. Okay, let's move to the next question. So, in humans, which hormone typically peaks in the early morning and helps promote wakefulness? So, I mentioned that there were two hormones. There are others, of course, but there is one. Yes, right. That's great. So, the answer is cortisol, which is option B. So, that peaks in the early morning, it promotes wakefulness. So, yeah. And there's another hormone, serotonin, which you may have heard of, that also peaks. I think typically it does. Actually, I'm not sure about peaking, but I know that it helps with wakefulness. Right. And the last question with this topic is which environmental cues, and these cues I learned are called Zeitgebers, and which of these environmental cues is the most important for entraining the circadian clock? So, regulating and making sure that the body knows when to do what. Yeah, great job. So, everyone's saying light, and that's the correct answer. That's option D. So as I mentioned, like the suprachiasmatic nucleus in the brain gets some input from the eyes as well with respect to light and dark detection, and that helps in training the circadian clock. And then that in turn talks to everything else and all of these other different regulations that are required. Right. So so these were the questions regarding the homeostasis. So now let's move to the next part. Does anyone have any questions regarding the stuff we just covered? Anything you're confused about? Anything you want to discuss? Let me know. You can also send a message if you don't want to talk out loud. Okay, great. So I'll move on to the next part, which was the bulk of the lectures, and I'm sure you guys really enjoyed the lectures. So this was about muscles. So we'll talk a little bit about the anatomy of muscles, and then we can see how you move from like a macroscopic structure to the microscopic structure to the molecular level of it as well. And we'll then talk about how muscle contraction happens and discuss some question answers in the meanwhile. In the meantime, right. So as we know, skeletal muscles that were mainly discussed in the work in the coursework. You start with, let me just get a pointer. Right. So you start with your muscles. The macro view of them is here. They can be attached via tendons, right, that attach it to the bones. And so if you look within the muscle, you see these individual bundles over here. And if you sort of zoom in on one of them, this is called a fascicle, right? And I'll go into the details of fascicle structure in a bit, but this is just the overview. So, and then you can see in the fascicle, you see these little round bundles over here in the transverse section, and each of these is a muscle fiber or a muscle cell. So those two terms can be used interchangeably. So each of these is a muscle fiber or a muscle cell. And these are essentially very long cells that have resulted from the fusion of many myoblast cells. So they share their cytoplasm. It's called a syncytium. So they're multinucleate cells that are very long and span the length of the muscle, right? And then if you look further into the transverse section of a muscle fiber or a muscle cell, you have individual myofibrils, okay? And these are these contractile structures that are actually contributing to muscle contraction. And the unit of myofibrils are what you saw in your coursework, which is the sarcomere. So multiple sarcomeres actually make a myofibril. So the sarcomeres are these repeating units that you see in muscle fibers. Sorry, in myofibrils. And so that's sort of the order of things: muscle, fascicle, muscle fiber or muscle cell, myofibril, and then the unit of that is the sarcomere, which ultimately is the unit of contraction. Okay. So now if we look at the more detailed structure of this, so here you have, this is the main muscle, and you see this covering of the muscle, right? So this is a connective tissue layer called the epimysium. So the epimysium covers the entire muscle. Now, if you look at the transverse section, you see these blobs which have little circular bundles in them. So these are your fascicles, right? So now we've taken out one fascicle over here. And this fascicle is actually covered by a perimysium. Okay, so that's a connective tissue that surrounds the fascicle. And then within the fascicle, you have your muscle fibers or muscle cells. Each of these fibers is further covered by a connective tissue layer called the endomysium. So you have three different layers of connective tissue protecting the muscle at its various levels of complexity: epimysium, perimysium, and the endomysium. You might look at this and say, okay, then what's the sarcolemma? So the endomysium is a connective tissue covering, and the sarcolemma is just the plasma membrane of the muscle fiber or the muscle cell. Okay. So further then, obviously, then you have the myofibrils, which are made of your repeating sarcomeres. And you have your innervation and your blood capillaries and blood supply, and then you have different organelles like your mitochondria and so on and so forth. You'll have your sarcoplasmic reticulum, very important for calcium release, all found within this region. And yeah, the nuclei, obviously. So yeah, then when you go to the structure of the muscles and you reach the point of the repeating unit of the sarcomere, oh, sorry. Yeah, so you have different types of filaments, right? So you can have myosin, which is a thick filament, and you can have actin, which is a thin filament, right? And they're arranged along these sarcomeres in a very specific manner that helps them slide along each other when we need to contract the muscle. So this is a sort of nice diagram that I found online where you can see the structure of the sarcomere. So as you know, the sarcomere consists of like the muscle cell or the muscle fiber consists of sarcomeres which have these dark and light bands. But one sarcomere is not one dark band and one light band. Specifically, it actually starts from between a light band and goes to the middle section of the next I band. Oh yeah, and I forgot to mention that the light bands are your I bands or isotropic bands, and your dark bands are your anisotropic bands. And the reason for them being called that is obviously one is that when you shine light, one appears light and the other appears dark. But what does isotropy and anisotropy mean is that if you shine light and it appears light in the isotropic band, it means that wherever you shine light from, the light passes through in the same way with the same refractive index. Whereas in the dark band, if you shine light from different angles, you have different refractive indices, which then cause it to appear dark, which is how it looks in a lot of microscopy images that you would have come across, I'm sure. So yeah, so moving ahead, here you can see that this is the I band, so the isotropic band or the light band. This is the A band or the anisotropic, then this is another I, and then so on and so forth, and it repeats that way. And here you have the Z disc. So this is in the middle of the I band. And what is the significance of the Z disc? Is that it is the place where actin filaments are, they come and they are bound here. So actin filaments connect between connect at the Z disc and they're anchored over there. And then they extend away from the Z disc towards towards the A band. And at the A band, you have your myosin. So myosin, this region, this H zone is a myosin only zone. And this, and in between, though it's not written here, there's an M line in between the H zone, right? Which you would have seen in the diagrams in your coursework. So in the middle of the H zone, there's actually an M line, and that's where all of the myosin is anchored. So the actin is anchored at the Z disc, myosin is anchored at the M line in between of the in the middle of the H zone. And then myosin extends this way, actin extends this way, and they meet over here, and that's where they overlap, right? So one sarcomere is therefore characterized as the region between two opposing Z disks. So you have half of an I band here, then you have an entire A band, and then you have half of another I band here until the Z disk, encompassing the M line and the Z and the H zone. Oh yeah, sorry, there was an M line mentioned over here, right? So so that's what a sarcomere is. I hope that's clear to everyone, right? And this is a little more of a sort of detailed drawing that's showing other proteins that are there. So the Z disk is actually a very an M line as well. They're actually very complex structures. It's not just one or two proteins. There's a whole bunch of proteins that are involved with anchoring actin, anchoring myosin, and so on and so forth. So alpha actinin here is important for anchoring actin. So actin is over here. And then you have titin, which was mentioned as well, right? It's a very flexible sort of spring-like protein that's very long, it's very big, and it's important for sort of moving across the entirety of this structure and acting as a spring for actin. You also have other proteins that I mentioned here. I'm not going to really go into details. Desmin is an important structural component of the Z disk as well. And myosin here is attached here as well. There's a protein called obscurin that's important at the M line, and so on. So this is the basic structure of the sarcomere and the corresponding regions in the I band and the A band, right? So, okay, so with this recap of what you've learned, let's go into some questions. So for anyone who's joined later, when we go through the questions, you can either unmute and say the answer, or you can send the answer in the discussion or chat box, and we can talk about it accordingly. Okay. So let's go through some more questions. So the connective tissue that surrounds an entire skeletal muscle is called what? So which of these is it? Do you remember? It's okay if you like you're not sure. You can just say anything. Anyone? I guess you don't remember, huh? Okay, so the answer here, the correct one. Oh, has anyone answered? Wait. um, Okay. All right. So actually, the answer is epimysium. So the outermost layer in the muscle is the epimysium that covers the muscle, the whole muscle. And then as you move inwards, you have the perimysium covering the connective tissue buffering the region of the fascicle, and then the endomysium, yeah, covering individual uh, muscle fiber. So yeah, so the answer here is epimysium. Okay. So a fascicle is best described as a bundle of which of these? Does anyone remember? So the right answer here is C, but I'm glad you're trying. Right? It's the muscle fibers. So the muscle fibers, or interchangeably known as the muscle cells, are bundled together inside a single fascicle. I can show you again, right here, right? So fascicles, individual fascicles, then you have the muscle fibers making up the unit of being the unit of fascicles, right? And this is the epimysium. Okay, right. So the next question is the plasma membrane of a muscle fiber is known as which of these? So this is a pretty straightforward one. Kind of it helps if you break down the word that you see, like, and look at the roots. Right. So the right answer here is B actually, but you're close. So anything sarco is going to refer to muscles, okay? And plasm typically is, the root word leading to something relating to like matrix within an enclosed region. So you have cytoplasm, which is, the matrix that all your organelles are suspended in any cell, right? So sarcoplasm would be the cytoplasm of a muscle cell, and sarcolemma is the plasma membrane covering of a muscle cell, because lemma is the root word that typically is associated with membranes. So the right answer here is B, sarcolemma. Okay, let's look at some more questions. So, which of these structures is multinucleated? So I talked to you about how certain cells fused together form something called a syncytium. So you have many nuclei, many mitochondria, uh, many common organelles that extend throughout this structure. So does anyone remember um, which one of these is the multinucleated one? Okay, so no problem. So the answer here is option C. Okay, so the muscle fiber or the muscle cell, because remember the muscle fiber or muscle cell is this super long cell that extends through the muscle, but it's not really originating from one single cell. It was a fusion event of many myoblast cells. They're sort of like the baby muscle cells, right? So they all fuse together and they form this syncytium or giant multinucleated cell, which is the muscle fiber or the muscle cell. Okay, so that's the one that's multinucleated. It's option C. Right. Another question is myofibrils. They're composed of repeating units. So what are these repeating units called? So I covered this just a little while ago. Do you remember? Yep, you've got it. So it's B. The answer is sarcomeres. So sarcomeres, composed of your A bands and I bands, are the repeating units of myofibrils. Okay, and they're also the chief contractile unit of the muscle. Okay, so now if you remembered, I didn't talk about this just now, but if you remember from your coursework, then that would be great. So T tubules, they run in what like orientation with respect to the sarcomere? So if imagine the sarcomere is like a flat line, then the T tubules would run in which way to that flat line, flat horizontal line? Would T tubules run parallel, perpendicular, both, or obliquely, which means at a particular angle? So, um, does anyone remember? We'll talk about T tubules a little more later with respect to their function when we're talking about muscle contraction. But for now, if you remember what was mentioned by your professor about T tubules, so that's her, right? So the answer here um, is A. Okay, so they're perpendicular. So they run perpendicular to the orientation of the sarcomere. So if you imagine the sarcomere as a line that's horizontal on your screen, the T tubules will run perpendicular to it. So they sort of invaginate into them, and then they are the ones that help conduct potentials. And so they're very important when neuromuscular signals need to sort of move across and allow for muscle contraction. And I think they're also important for distribution of certain materials as well. Yeah. So I guess it's like you have one really long cell. How does it communicate? So T tubules help do that. Okay, great. So let's move on. I think I have a couple more questions and then we'll talk about muscle contraction. So the Z line here defines the boundary of what? Which part? Right. So one answer has come in D. Does anyone have any other answers? An answer has come in C. So actually the answer is B, and I'll explain why it is the sarcomere, right? So let's go back to this diagram. Sorry, I think this one is good, right? So this is the I band, this is the A band, right? This is the Z disc. So you have one Z disc over here and another over here. So a sarcomere is what is defined as the region encompassed or enclosed between two Z disks or Z lines, right? So, I mean, you're right in saying that H zone is there in between two Z disks. That's not technically wrong. But when it comes to the functional sort of definition of it, the right answer would be sarcomere. Okay, so sarcomere is what's enclosed between two Z disks. Yeah. So the Z line defines the boundary of the sarcomere. Okay, next question. So which protein anchors actin filament at the Z disc? It's not the only one, though I have to amend this question a little. So it's not just this protein. There are other proteins as well that are part of anchoring, and this is one of them. Does anyone want to try? So troponin, troponin anchor. I believe troponin does not anchor at the Z disc, unfortunately. It binds to actin, like troponin is bound to actin, right? You have those different subunits of troponin that are bound to actin and tropomyosin, and that's important for myosin binding, but it's not what anchors. So one of the anchoring proteins actually is titin. I think also alpha-actinin, these are all involved in anchoring actin filaments at the Z disc. So the right answer here would be C, titin, and others. Right. Okay, great. So next question. So the A band or the anisotropic band corresponds to the region containing which of these? So I showed that diagram earlier where there were a certain distribution of actin and myosin filaments. So, does the A band have only actin, only myosin, an overlapping region, or all of myosin? Maybe think back to what you know of sarcomere structure, and that might help you decode this one. Anyone? So maybe I can explain again with the sarcomere structure. So, right. So the A band you can see is this region, right? So if the answer were only myosin, that would actually correspond to this region. So the H zone has only myosin, right? The blue is myosin, by the way. And the Z disc region has only actin. This region in between, extending between the I band and the A band, this has both of them. And the M line and H zone, this region has only myosin. So if we were to talk about the A band, that actually contains all of the myosin. So the entire extension of myosin is contained within the A band. Okay. So the right answer here was D option, which is the entire length of myosin. I hope this is clear. Like the sarcomere structures. Anyone confused about anything, please stop me and ask me, okay? All right, last couple of questions. So, which region appears light under a microscope due to thin filaments only? So this one should be clear by now, I think. So it would be great if someone answered. Oh, uh, so so you have to remember what the sort of names mean over here. So the A band and the I band are really the two options that you need to care about here. So A band, which is the anisotropic band, that's the one where you have light that reflects sort of in different refractive indices when it's shown from different directions. So it actually A band is the one that appears dark under the microscope, whereas I band, the isotropic band, where if you shine light from different directions, it actually has the same refractive index, so it appears light in under the microscope. That's the one, and also why is that? Because I band contains thin filaments, which is actin, whereas A band contains thicker filaments, which is myosin. Okay, so here the right answer is C option. That's right, yeah, C option, which is the I band is the one that appears light under a microscope. So when you see this filament, the muscle filament under the microscope, you see alternate dark and light bands. The dark one has more myosin, and that's the A band, whereas the light one has more actin enrichment, and that's the I band. Okay, so I hope that's clear now. Right. Now the M line that we talked about, right? Um, Primarily contains proteins involved in which of these functions? So just to remind you, the M line runs through the H zone. So the H zone is in the A band, right? So I hope this isn't too confusing. So the A band or the dark band, which contains, and I'm going to give a big hint here, but the dark band that contains thick filaments, okay, so the A band contains the M line in it, right in the middle. So what role do you think the M line could be playing over here? Right? So the M line, which is in the H zone, that is a place where there's only myosin. Does that help? If there's only myosin, which of these answers could it be? Right? So it's option B, right? Myosin anchoring. So that's the right answer here. So the M line, which is running through the middle of the A zone, the A band, is where myosin is anchored, and it's just myosin over there. So one way to remember this is M line, M for myosin. That's how I remember it. So you can try to keep that in mind that M stands for myosin. So there's only myosin there, and myosin is what's branching outwards from there. So it's anchored at the M line. Okay, so the proteins that are involved in this process are primarily found in the M line for myosin anchoring. All right. Another question is which elastic protein connects the Z disk to the M line? So the key here is to look at the word elastic. So do you remember which protein particularly was mentioned to be highly elastic? Yes, exactly. So the right answer here is D option. Good job. It's titin, which is it's like a coiled spring-like protein, and it extends from the Z disk, or between the Z disk and the M line. So again, maybe we can look at the structure. So you can see here is the Z disk. All the way across, you have the M line, and then obviously you have more of the sarcomere. So it's extending all the way. It's humongous protein. Okay. And it's elastic. It helps with sort of maintaining the stretch and bringing things back to equilibrium, like any spring would. Okay, I think these are the last three questions before we move to the final topic. So, which of these structures gives skeletal muscle its striated appearance? So the key here is to know what striated means. Anyone? So, unfortunately, it's not A. The answer here is B, right? Because sarcomere organization is what gives it the striated appearance. Sarcomeres are made of alternating light and dark bands, or I and A bands, right? So, because you have this alternating arrangement, um, of light and dark bands, that's what gives it a striated appearance. So striated essentially means just that, right? So these skeletal or voluntary or striated muscles, they're called striated because they have um, this pattern of alternating dark and light. So that's what striated means, and that's and this appearance is caused by sarcomere organization having alternate light and dark bands. Okay. Excuse me. So, all right. So the next question. So now, think back to what you've learned about tropomyosin and troponin and what I just mentioned a little earlier. So where is troponin located? Okay. So where is troponin, located? So I mentioned what it is bound to. So the person who just joined, you can write the answer into the chat, or you can unmute and say the answer out loud, whatever is your preference. And we're discussing them as we go. Okay. So, right. So the answer here is the thin filament. It's located on the thin filament because you have to remember the thin filament is actin. Okay. So actin, which is made of it's a polymer of G actin, right? So G actin are the individual building blocks that make a filament of actin, a filamentous actin, right? So you put many G actins together and you get a filament, right? And the troponin and tropomyosin, I'll just show you from the image. Uh, Maybe it's not visible here. Huh. Yeah, I don't think I have an image for it. But essentially, if this is actin, and you see this sort of helical structure, this is actin. Okay, so you have these actin dimers that come together and form this polymer of filamentous actin. And tropomyosin, if you follow the pointer, it's essentially just bound along actin, and that's what, and it binds to each and every spot on G actin where a myosin head can potentially bind. So it blocks those sites, right? And troponin is bound right next to it. So it's on the thin filament or it's on actin. Okay, right. Yeah. Yeah, so the right answer here is B, thin filament. Okay. Now, which of the following is not part of the thin filament? So what is the thin filament? If you know that and you think about the structure that I just showed you, then you'll be able to know which is not part of the thin filament. Yeah, exactly. So the right answer is D, as you mentioned. It's myosin, right? Because myosin is the thick filament. So it's not part of the thin filament or that assembly in general. Actin is the thin filament, or the globular or G actins coming together to form a polymer of filamentous actin. That's a thin filament. And tropomyosin and troponin are bound onto actin. Myosin is in the thick filament, which is separate. All right, so these were 15 MCQs. I had this one question that I thought might sort of summarize what we've learned so far with respect to macro to micro organization. So just think about this one and let me know what you think the answer is, and then we'll discuss it. So the correct order for the smallest to largest unit of organization in muscle tissue in general is which of these? So smallest to largest from left to right, which one do you think is the correct order to place these? So take a good look and let me know what you think. And then we can discuss. Try to remember what we've learned so far about muscles, about fascicles. Okay, so I've got some answers in now. So let's see which one is right. So the smallest unit is a myofibril or a filament. So that's one of the options here, is maybe there could be a filament, right? Then the next unit is a muscle fiber or a muscle cell. So where this one is gone. So it's not D, unfortunately. After a muscle fiber or a muscle cell, you bundle them, and you have many of these muscle fiber bundles, which forms the fascicle. So fascicle is in number 3. So which of these has fascicle in the third position? That's option B. And then many fascicles come together to form the complete muscle. So the answer here is B. You start with a myofibril, which are basically the long fibers made up of sarcomeres, right? And actin and myosin, they all come together to form these long contractile cords within within the cells, and each of these make up one cell, which is a muscle fiber or a muscle cell, and they come together, many of them, to form a fascicle, which then or which this is a single fascicle, and then they come together to form the muscle. Okay, so the correct answer here is option B. So hopefully that's clear, and if that was a confusion before, I'm hoping that that's cleared up now. Okay, so now you have this to refer to. Okay, so what is this, right? This is a sarcomere, right? I mean, it's depicting a sarcomere and some A bands, I bands, and so on. So looking at this, I wonder if you're going to be able to do a small mathematical exercise that I have prepared for you here. So imagine that you have a length of myofibril. So myofibril is what? Myofibril is this length of actin and myosin and and repeating units of sarcomeres that are extending across the entire muscle. Okay. So many sarcomeres make up a myofibril. Okay, so if I have a length of myofibril that contains 5 sarcomeres, imagine that the myofibril is this long dupatta. Okay, and you have a repeating pattern in the dupatta. So essentially, I have some X length of myofibril that of this myofibril dupatta that contains 5 of these patterns, right? And this pattern is what? It is this between these two Z's. This is the pattern, which is your sarcomere. So I have 5 sarcomeres back to back. If I have that, in these 5 sarcomeres, how many H zones will I have? So think about it. You can use this as a tool for visualization, and you can count and tell me in the chat how many H zones will be there in 5 sarcomeres. Just think about it, right? You can use this, right? So maybe I'll help with this one, and then the next one might become easier to do. So if you see this diagram, I mentioned that the sarcomere, right, extends between two Z lines, right? So you have a Z here and a Z here, and everything in between is the sarcomere, right? So all you have to do is figure out how many H zones are there in one sarcomere and then multiply by 5, right? So in one sarcomere, you have one H zone, right? That goes right down the middle. So this is the A zone, and the H zone or H line is right in the middle um, of the A band, right? So for every sarcomere, you have one single H zone. So for 5 sarcomeres, you are therefore going to have 5 H zones. So the right answer here is 5. Okay. All right. So the next one is how many Z lines will be included in 5 sarcomeres. So I'm doing this exercise just so that it's clearer what really a sarcomere is and what its constituents are, right? So I'm hoping this helps if there's any confusions. So how many Z lines will be included in 5 sarcomeres? So again, envision this one and see how many are there in a single sarcomere. So I've got one answer in, and you are absolutely right. It is 6. Good job. So see this one. This is one sarcomere. You have 2 Z lines. This is another sarcomere, another one. So you have 4 now. Now imagine, because this is just three sarcomeres, 1, 2, and 3, imagine that there's one more here and one more here on the left. So that would be one more sarcomere, one more Z line here, and one more Z line at the boundary over here. So 1, 2, 3, 4, 5, and 6. So enclosing 5 sarcomeres are 6 Z lines. So the answer here for B is 6. Okay, so good job. Now, I'm sure you'll get this one. How many A bands will be enclosed in 5 sarcomeres? Yeah, exactly. There will be just like H zones, A bands, there will be 5, because a complete A band is enclosed within a sarcomere, whereas it gets a bit more, you know, dicey with the I bands because there's like half here and half here and so on and so forth. So A bands are completely enclosed per sarcomere between the Z lines. So for 5 sarcomeres, there are going to be 5 A bands and 5 H zones and 5 M lines, which are the ones that divide in right in between the H zone. Okay. All right. Last one is, how many regions with only myosin and no actin will be there in 5 sarcomeres? So for this, you need to remember like which region actually has only myosin and no actin, right? So we discussed this earlier, but I can go through it quickly just now from this diagram itself. So if you remember what I had mentioned, actin is anchored at the Z disc, okay, or the Z line, and it extends outwards from the Z line in both directions. So this black thing, this black line is actin, okay, and this yellow is what is anchored at the M line or the H zone. Okay, so M line is just in between the H zone. So whatever it is, the myosin, which is the yellow color thing over here, that is anchored over here, right in the middle of the H zone at the M line. So that means, and this is the region of overlap between actin and myosin where maximum cross bridges will form. So in a resting muscle, this region has only myosin. So your H zone has only myosin, right? So this question regarding how many regions with only myosin and no actin actually just is the same as question A, which is how many H zones are there, right? So the answer here is 5, right? So there's 5 H zones in 5 sarcomeres, and so there are 5 regions with only myosin attached at the M line and no actin. Okay, so I hope that then now cleared up any of the concepts relating to sarcomere structure, muscle structure, and so on. Okay, so if the anatomy is clear, then it's easier to then understand what's going on in the functionality, right? Which is the muscle contraction that takes place. So, if anyone wants to ask anything else, let me know. If you have any confusions, please let me know before I move on. Is everyone good? Yeah, okay. All right, then let's move on to muscle contraction. So I thought instead of me, going over the same diagrams that you've already seen, I found this really nice video on YouTube, which I thought I'd show you guys for reference with respect to how the basic muscle contraction phenomenon takes place at the molecular level here, right? So with respect to actin and myosin and tropomyosin, troponin, and how there is and the importance of calcium and ATP in this process of muscle contraction. So how this pulling between the muscles between actin and myosin takes place, right? So I've also included the link in case you want to watch it separately for yourself later on. So I'll just play the video. If you can't hear it, let me know, okay?
In this video, I'll introduce you to the tiny proteins, myosin and actin, that are responsible for helping your skeletal muscle cells contract. I'll review the series of steps required to make this contraction happen, often referred to as the cross-bridge cycle. You ready? Let's do it. Welcome to PhysioFlip. To get a good sense of how tiny proteins in our skeletal muscle cells cause muscle contraction, let's first zoom in onto a small section of the sarcomere, the structure in skeletal muscles that is responsible for generating force. In this sarcomere, we can see a variety of different proteins. Here we have the thick filament, composed mainly of the protein myosin, and the thin filament, composed mainly of the protein actin. Along the thin filament, we also have a couple other proteins, including tropomyosin and troponin. These four proteins together will help control if or how much force this muscle fiber's sarcomere can generate. To generate force in our muscles, myosin has to bind to actin. It has to grab it, pull on it, and shorten the sarcomere. However, myosin normally cannot bind to actin because the protein tropomyosin is blocking access to actin. You can see the binding sites for myosin here on actin being blocked by tropomyosin. That's why I kind of like to think of myosin and actin like two teenagers who want to date. Myosin just wants to go out with actin, but there are these parents, tropomyosin and troponin, who are blocking that, preventing it from happening. But for this muscle to contract, we need myosin to bind to and pull on actin. So let's go through the steps that allow this myosin to bind to actin and eventually pull on it to generate force. First, myosin will bind ATP and break it down or hydrolyze it into ADP and an inorganic phosphate molecule. The energy released from this breakdown of ATP is then used to push the myosin back into position to an energetic state so it's ready to bind to actin. But tropomyosin is still in the way. The parents are still blocking myosin from dating actin. Well, that's where calcium comes in. Calcium can bind to troponin, activating both troponin and tropomyosin, which causes tropomyosin to change shape and expose the binding sites for myosin. That's why releasing calcium from the SR was so important. Without calcium, myosin could never bind to actin and would never generate force.
Oh, sorry. My bad.
The energy released from myosin will now bind to actin, and by releasing the
Sorry.
activating both troponin and tropomyosin, which causes tropomyosin to change shape and expose the binding sites for myosin. That's why releasing calcium from the SR was so important. Without
So this is talking about the importance of calcium, right? So I thought I'd just talk about few things, right? So one thing is, what is the SR? The SR is the sarcoplasmic reticulum. So that is essentially the endoplasmic reticulum of the muscle fiber, and that contains a lot of calcium. So if the cell suddenly requires calcium to allow myosin binding to actin, where does this calcium come from, right? So we have a store of it in the sarcoplasmic reticulum, and it is released from there, and then it comes and binds to troponin. So where does it bind, right? So it binds to tropopin C, which is the site which binds calcium, and then that leads to certain changes in the geometry of these binding elements, and that allows for the myosin head to then bind because it exposes the region um, and prevents tropomyosin from blocking that region on the G actin. And so then it allows the myosin head to come and bind. So SR is the sarcoplasmic reticulum, and the region actually of the one of the three subunits of troponin that is bound by calcium is troponin C. Okay, right. So I just thought I would
Without
reiterate that.
calcium, myosin could never bind to actin and would never generate force. Now, with tropomyosin out of the way, myosin will now bind to actin, and by releasing the phosphate, generates the power stroke, which is just a fancy name for saying that myosin actually pulls on actin and generates force. At the end of the power stroke, the ADP molecule is also released, and the myosin will stay attached to actin in a state known as rigor. If we want to generate even more force, we need to just repeat this cycle. That will happen when a new ATP molecule binds to myosin, releasing the myosin from the actin. The ATP is then broken down just like we saw before, with the energy being used to push back the myosin and get it back into the ready and more energized state. If calcium is still present, myosin will bind to actin again and repeat the process to generate even more force. Let's quickly recap the steps of this process, often called the cross-bridge cycle. First, myosin breaks down or hydrolyzes an ATP molecule, using that energy to push the myosin back into a more energetic state and get it ready to bind to actin. Myosin will then bind to actin if calcium has been released from the sarcoplasmic reticulum. Myosin then releases the phosphate group, pulls on the thin filament, and slides it towards the center of the sarcomere, going through the power stroke and generating force. Next, myosin releases the ADP molecule, forming an even tighter bond with actin and entering a state known as rigor, in which the myosin has pulled on actin, is generating force, but is still stuck to the thin filament. To release myosin, either to help the muscle relax or to get myosin ready to start another cycle to generate even more force, myosin will bind ATP, releasing actin, and allowing us to start this process all over again. That's a lot of steps, right? But here's the result. Myosin will pull on actin and shorten the sarcomere to generate force. By the way, you might notice that neither actin or myosin filaments are actually getting shorter. All that happens is that actin slides over myosin, being pulled towards the center of the sarcomere, shortening the sarcomere to generate force. That's why this process is often called the sliding filament theory of muscle contraction, since the thick and thin filaments seem to slide over one another.
All right. So I hope that was informative. I thought it was a pretty clean demonstration of the sliding filament theory. So yeah, with that, let's move on to some questions. And we can discuss them as we go in case there's anything you're confused about. So this is literally just what we saw. So muscle contraction is best explained by which of these theories? Could you tell me which one you think it is? Yes, exactly. So the right answer is option C over here, which is the sliding filament theory. It's, follows from the name that you have a filament sliding with against another one, pulling it, right? So sliding filament theory, given by Huxley and I don't know how to pronounce it, Niedergate, right? It's a really important theory that talks about how muscles contract using thick and thin filaments. And they don't actually, the filaments don't actually contract, but they slide along each other. And so your A band doesn't actually, like when myosin is sliding, your A band is actually not changing in its length. Instead, you're changing and shortening other regions, which we'll talk about. Right. So the immediate trigger for muscle contraction is which of these? So, hmm. So the answer here I had sort of written down as B. So essentially, calcium binding to troponin, that's what frees the binding spot on actin for myosin binding, right? Because without that, there is no muscle contraction. So calcium is honestly like the very first and most important thing when it comes to muscle contraction. So calcium binding troponin frees up the site that was initially blocked by tropomyosin and allows for myosin to bind to G-actin. Okay, so the answer here is B. All right. Um, Calcium is released into the sarcoplasm, which is nothing but the cytoplasm of the muscle fiber. From which of these? So I actually mentioned it during the video. I wonder if you remember. Yes, exactly. So it's the sarcoplasmic reticulum, which is nothing but the endoplasmic reticulum of the muscle fiber. So it has a huge store of calcium, which is and as was talked about later, there are channels there and ATP gated pumps that essentially maintain this gradient of calcium where you have a highly concentrated calcium within the sarcoplasmic reticulum and relatively less concentration in the cytosol. And this gradient is maintained very strategically with the help of these ATP pumps, where I think every ATP hydrolyzed results in two calcium ions being pumped inside the sarcoplasm against the concentration gradient. That's after the signal for contraction has been withdrawn. So if you remember from the last part of the lecture where we talked about the neuromuscular junctions, once you have acetylcholine esterase that clears up the acetylcholine, so you don't have any of that signal for contraction anymore. You actually need to take that calcium back into the sarcoplasmic reticulum. And so that's when this released calcium from the sarcoplasm is transported back into the SR with the help of ATP hydrolysis. Right. So, yeah, so anyway, that was an extra thing, just like a reminder. Okay. Yeah, so let's move on to the next question. So question 19, binding of calcium to which troponin subunit initiates contraction? So I mentioned it earlier on as well when I had stopped the video. So there's an easy way to remember this one. So the answer is unfortunately not tropomyosin, though I can understand why one might think that is the right answer. So it's actually troponin C. So you can remember C for calcium, right? So troponin C is the one that binds calcium. So troponin has three subunits: troponin C, troponin I, and troponin T. Okay. So troponin T, which is I think you can see my mouse. So troponin T is the one that binds binds tropomyosin. Okay, so troponin T, so troponin itself is really close to tropomyosin, and tropomyosin is directly bound to actin, and it blocks it. Right, so troponin T is the one that binds to tropomyosin. So T for tropomyosin. Troponin T is the subunit that binds tropomyosin. Okay, so that's what keeps it in place next to tropomyosin. Troponin I is the one that binds to actin. So you can remember it as I being the isotropic band and the isotropic band containing actin, right? So the thin filaments in the isotropic band um, um, are what make it. So troponin I, which binds to actin, that's the way you can remember it. Okay, so this one, it acts like a club and it binds actin. Troponin T binds tropomyosin. So that's what keeps troponin in place between tropomyosin and actin. So finally, that leaves troponin C. third subunit, which is C for calcium. So that's where calcium binds. And once calcium binds there, it triggers some sort of conformational changes that allows tropomyosin to no longer block the G-actin subunit, the G-actin region where myosin binds. Okay, right. So I hope that is clear. All right. Okay, question number 20. So ATP binding to myosin causes which of these things to happen? So I hope you remember what you saw in the video or what you've read in the coursework uploaded, right? So myosin is a molecular motor, right? So it literally converts the energy that it generates from ATP hydrolysis to mechanical, like actual, there's actual displacement, there's actual work done that occurs, right? So if myosin is bound by ATP, so the answer here is actually cross bridge detachment. So it's option C. So when myosin is bound to ATP, actually it doesn't form the cross bridge. When ATP is um, hydrolyzed into ADP and phosphate, inorganic phosphate, that's when the cross bridge detachment takes place. Right. So ATP bound myosin is when it's not bound to the cross bridge, where bound via the cross bridge, and when you have ATP hydrolyzed to ADP and phosphate, you have the vice versa condition, right? Right. So I hope that's clear. It was explained in the video. Right. So now, the next question is about the power stroke, right? So once you've already bound um, myosin and actin, how does this power stroke occur? What is exactly the step that triggers the power stroke? So it follows from the previous question, right? So if you have ATP bound to myosin, so you have cross-bridge detachment. So that follows that if you have cross-bridge formation, that will lead to a power stroke, right? Because that means myosin is bound to actin. So if you have an ADP and phosphate being released, we know that cross-bridge formation has occurred, which will then be closely followed by a power stroke. A power stroke is nothing but myosin sliding along and pulling actin. Okay. So the answer here is option B, when ADP and inorganic phosphate are released. Okay. All right. So let's go to the next question. So to the person just joined, you can send the correct answer that you think into the chat, or you can unmute and say the answer out loud. And we'll discuss as we go. So we're talking about muscle contraction right now and some questions relating to it. Okay. All right. So this question is, during contraction, which of these regions, which region listed below actually shortens? So does anyone know which one it could be? There's some regions that don't shorten, which was part of the sliding filament theory, and some region where the length does change. So any idea, anyone? So I have an answer here, A. So actually, A band is the one that doesn't shorten. Okay. Because you have to remember that myosin, as part of the sliding filament theory, is actually not like the thick and thin filaments themselves are not contracting but sliding along each other. So the filaments themselves are not becoming shorter or longer. It's just that these particular regions that are defined by the lengths of these filaments, defined by the extent of these filaments, that's what's changing. So if you have them sliding along each other, what would happen, right? If you remember the structure of a sarcomere, right, the A band is constituted by myosin only. And if the myosin is actually not changing in its length, then the A band stays the same. What changes actually, and the answer here is the H zone. I think the I band also does change considering actin is sliding. So the sarcomere is becoming shorter in general, but the A band stays exactly the same length, whereas the I band uh, becomes contracted. So the I band and the H zone are actually what change in their length and they shorten during muscle contraction. The A band stays the same. Yeah. And the Z line and M line are just regions of attachment, so there's nothing really with respect to them shortening. Okay. All right. Next question. So during relaxation, so opposite of contraction, calcium is actively pumped back into which of these? So I was just talking about calcium and how it's regulated from in the during contraction. So remember that calcium comes out of a certain organelle when it's needed during contraction, and then it sort of goes back there in an active mechanism. So active means that ATP is consumed. Yeah, exactly. So the answer is C, very correct. Sarcoplasmic reticulum. So that gradient and that concentration difference between the sarcoplasmic reticulum having high calcium and the sarcoplasm having a much lower calcium concentration is maintained so that once the muscle is relaxing, essentially in a neuromuscular junction, if you have acetylcholine being degraded by acetylcholinesterase and there's no longer a signal to contract, the muscle starts to relax and calcium is actively pumped back in with the help of an ATPase into the sarcoplasmic reticulum. All right. So the answer here for question 23 is option C. Okay. All right. Now, which enzyme activity is intrinsic to the myosin head? So this should follow from what um, you learned earlier from the video. Yep, I think you guys are getting it. Great. It is, yeah. Does anyone else want to try? Yeah. Yeah, so it's option C. It's ATPase. So we know that the myosin, it's a molecular motor, and it, binds ATP, it hydrolyzes ATP. So that's the job of an ATPase. And so the enzyme activity that is intrinsic is essentially that intrinsic means that it's just the property of something. So it's the property of the myosin head, and that's ATPase enzyme activity, right? So, and that's highly important for regulating when it binds, when and if it binds to actin, right? No, so the answer is not kinase. Uh, uh, um, so I just explained it, so I guess it's clear now. So it's not kinase, it's ATPase activity. So kinases remove phosphates. So one might say that, okay, yeah, that's that's what's happening here in ATP. But yeah, so the answer here is ATPase. Okay, last couple of questions. So this was mentioned in the coursework. It was super interesting, right? Rigor mortis occurs due to the lack of what? So if you remember what rigor mortis is, right? It's like if mm, it's like if a person has passed away and is dead, then soon after the death, the muscles seize up and they're very stiff. You'd see in a lot of these crime dramas, they'd say, okay, so rigor has set in. So that means that we can place the time of death at XYZ hours before rigor set in, because you can predict from when rigor mortis sets in when the person actually died. Interesting. Yes, exactly. Right. So the answer is ATP, which is option D. Good job. Um, yeah, you're absolutely right. So when someone is died, their cells are no longer metabolically active. There's no more ATP being produced. So there's nothing to replace the myosin head with. So when ATP is bound to the myosin head, that's when it is detached from actin, right? But if there's no ATP being actively produced, then you don't detach, and the myosin is stuck, and it's in the form of rigor. So it's very stiff. But then after a while, of course, things start degrading, and then the body becomes limp again. So that's different. But yeah, there's a period of time when there's high rigor. Okay, so next question. Which event directly follows acetylcholine release at the neuromuscular junction? So this one we have discussed, so it should be hopefully relatively clear to you now. Right. So the answer here, that's absolutely right. It's option C. It's not A, it's C. So when acetylcholine is released at the neuromuscular junction, and it fuses with the muscle membrane, sorry, it it interacts with the receptors on the muscle membrane, right? The acetylcholine receptors. That leads to depolarization of the sarcolemma, which is propagated via the T tubules. Oh, I think I gave away an answer. Oh. So this depolarization, essentially a change in the extracellular membrane charge, is what immediately follows. And then once that reaches the place where the membrane is interacting with the calcium channels in the sarcoplasmic reticulum, that results in calcium release. So the first thing after acetylcholine is interacting with acetylcholine receptor is depolarization of the sarcolemma that propagates and then leads to calcium release. So calcium release is one or two steps beyond this. Right after this is option C. So you're absolutely right. Uh, let's go through the last couple of questions, and then we will be done for today. So this I just gave away, but the T tubules, they primarily function to do which of these activities, which of these functions? So I was just talking about how Acetylcholine binds to acetylcholine receptors found at the neuromuscular junction, and that leads to depolarization. And that's not just at one spot, it actually needs to be transmitted, right? So the correct answer here is option A. T tubules, that are the structures that are sort of membranous invaginations that are perpendicular to the direction or the orientation of sarcomeres. These T tubules are very important for transmitting action potentials that are generated at the NMJ, or the neuromuscular junction. Okay, so I hope that's clear. I hope you remember this now. Let's move. These are the last three questions before we're done for today's session. So, increased muscle tension could be achieved by which of these options? Oh, sorry. Yeah, that's right. It is option B. It's recruiting more motor units. That's exactly right. So what does that option even mean, right? So recruiting more motor units means that you have more myosin that's bound, like more myosin, and essentially that more myosin will bind to actin. So motor protein here is myosin, and myosin binds to actin only when there is enough calcium to allow this, because as I mentioned and we have discussed quite a bit now, that tropomyosin, it blocks actin, and to remove it you need calcium binding to troponin. So option D, it cannot be, because reducing calcium release would literally do the opposite of that. Decreasing ATP availability would just cause the myosin to get stuck on actin and not detach. Increasing sarcomere length makes no sense, because that would actually, if you were able to like stretch a sarcomere somehow, then that would actually lead to less interaction of myosin and actin. So the only viable option here is option B, which is recruiting more motor units. So recruiting more myosin, or sliding more myosin such that it interacts with more actin. So optimal positioning allows for more recruitment of motor units, which can increase muscle tension more, because there'll be more contractility. Okay, so this one directly sort of links to the previous question, which is which condition results in maximal cross bridge overlap? Okay, so okay. Yeah, it is optimal sarcomere length. So the reason it's not shortened sarcomere is you have to remember that sarcomeres don't really like wholly shorten, like specific regions shorten at times, but the if the whole sarcomere were to shorten, nothing would change, right? It's specific regions have to shorten at specific times. So actually, there's an optimal sarcomere length where your H zone and I band are reduced in length, that allows for more interaction of motor units like myosin with actin, that allows for maximal or optimal cross bridge overlap. So the answer here, though I understand it's a bit confusing, I admit, is option A, which is optimal sarcomere length. So you're right. Okay, and the last question for this session is relaxation occurs when which of these following options? Yeah, so you've got it right. Yes, both of you have got it right. That's great. So it is option C, which is tropomyosin blocks the